disease that I am presenting today is the bipolar disease by me, Shalissa Widow, with the dopamine effect. What is this disorder, you may ask? It's also known as bipolar affective disorder or manic depressive illness. It causes unusual shifts in mood, energy, and activity levels. Manic episodes are caused by periods of abnormally and persistently elevated mood. Behavior may lead to social or occupational impairment. Major depressive episodes are characterized by a depressed mood or loss of interest or pleasure in usually pleasurable activities, and extreme and opposite symptoms like weight loss or gain, insomnia or hypersomnia, psychomotor agitation or retardation, fatigueness, or the feeling of worthlessness or guilt. The receptor protein is a G protein coupled cholinergic receptor, which is also known as GCPRS, which has one binding site, and the binding site could be either extracellular or cytoplasmic, as agonists that bind from the extracellular space, termed the orthosteric site, promotes binding to another partner at the cytoplasmic side. GPCRs are diverse and bind with many different types of ligands. Characteristics unique to the signaling pathway. There's alterations in the CAMP26, another second messenger system involving protein kinase C signaling 27, and the phosphodiesterase pathway, in which protein kinase C and GSK3B are second messengers. WNT binds to FZD and LRP56, thereby recruiting the scaffold protein disseveled DVL to the ligand receptor complex. And when I show you the normal pathway drawing, you will see more of this, especially the scaffold protein, which comes in the end at the ECM. Protein kinase C is involved, in which increases during maniac settings, and it controls the function of other proteins via the phosphorylation of hydroxyl groups of serine and threonine amino acid residues. The G protein involved is the GPCRS. Pathway that leads to the bipolar disorder, which again, it's going to be more detailed when I talk about the drawing. Cell surface, but it starts with the cell surface receptors to induce extracellular signals such as neurotransmitters and neuropeptides into the interior of the cell. Those transduced signals communicate with other cells by activating 7 transmembrane spanning G protein coupled receptors. GPCRS activate G proteins, which are composed of A and BY subunits. 2 families of proteins turn off the GPCR signal, and then uncoupling the GRKS phosphorylate GPCRPs from the respective G proteins. The GTPase activating proteins accelerate the G protein turn-off reaction, and the 2 major signaling cascades that are activated by GPCR are the CAMP generating second messenger system and the PI system. Then the CAMP goes on to activate the PKA, and then the TRPM1 channels are downstream of the MGLUR6 receptor. And as you can see here, this is what the normal pathway would be, normal signaling pathway would be. And as you can see, it gets much more complicated in an abnormal pathway. Cell response termination. A nuclear response is regulated in all genes except one, the cardiac B adrenic signaling involve nuclear gene regulation. And there's many ways to terminate the signal, but the one way I found was to terminate the signal is to remove the ligand from the GPCRS receptor. References, and this is one of the references where I got most of my sources from, but I would also like to acknowledge the biology department and Dr. Byers for the information that they gave us. And then I'm just going to go on to talk more about my drawings. As you can see here, this is what a normal pathway would be. The ligand binds to the receptor, and you get into your cellular response. But then, as you see, when it comes to an abnormal pathway, it gets more complicated with the dopamine effect and everything. And as you can see, it ends up in a long-term alteration of the synaptic plasticity, and it affects the nucleus. And terms that are well recognized would be, which I talked about in the presentation, would be the CAMP, which the CAMP goes on to activate the PKA, but it goes on to more of effect. The PLCB activates the IP3, and the CA plus goes on to activate PKC. And then this over here was a scaffold protein, gisivald, which I talked about at the end, which really comes from the WTS pathway, which is a lot of different, as you can look in this signaling pathway, there's a lot of pathways that interact with each other, that intertwine with each other, which makes it more complicated. But this is what it looks like when you have a bipolar disorder, and this is what the signaling pathway would look like. And I try to make the abnormal pathway look a little abnormal. That's why it looks like a D, but it's supposed to be upside P because it's abnormal. Thank you so much for watching.